Welcome to the Medicines for Africa webinar series, which is called Building Africa's Frontlines Against Unsafe Medicines. And we have a series of these webinars happening every 2 weeks, starting from the 15th of April. And today's session is going to focus on the critical issue, emerging issue of nitrosamine contamination. And our speaker is going to be Dr. Partha Mukherjee, the Executive Director for Analytical Chemistry at Lilly. And I will introduce him in a moment, but perhaps I introduce myself first. I'm Lenias Hwenda, and I am the founder and CEO of Medicines for Africa, an independent Pan-African social enterprise that is dedicated to protecting patient safety by improving medicine safety. Our mission is to protect patients from harm, medicines-related harm across African markets. And our vision is to become a trusted partner of choice on patient safety across the African continent. Patient safety has never always been front and center in the African setting. And Medicines for Africa, we are working with the purpose to change that. Not only to put patient safety on the agenda, but to drive the actions that are needed to make medicine safer for everyone. With that, I would like to, so we already had a webinar a couple of weeks ago where we had Dr. Martins Iluyomade from NAFDAC. And he basically gave us our inaugural webinar talking about the challenge of unsafe medicines and which used to be a hidden challenge, but perhaps not anymore. So I'm going to pass the floor over to my colleague, Lydia Bisecton, who is going to just give us a recap of that webinar. Lydia, if you could come in and just tell us what were the things that really struck you from or the takeaway messages for you, your personal reflection, just to give people who weren't there a recap of the conversations we've had or what's happened, which we are going to build on today before we bring in our speaker. Over to you, Lydia.
Thank you, Lenias, first and foremost for the introduction, and good day, good evening to you as well as all our participants. So before I even begin with my reflections, could I please just have a show of hands with regards to who was present for the session that we had with Dr. Iluyomade? Just so that I know whether I'm speaking to people who participated in the last session or we're speaking to new participants. You can just raise your hand or react. Okay, so I see David was there for that session. Are the rest of us new participants? So if you're not there, maybe react with the waving or the clapping emoji, just so that I see. Okay, I see Snetemba is sending the clapping emoji. Let me just give a minute for everyone else to show before we begin with our reflection. Okay, so it looks like we have a mix of people who may have attended or who may have not. But essentially, as Lenias earlier on introduced, the last session that we had was with Dr. Martins Iluyomade, who is the Director of Investigation and Enforcement at Nigeria's National Agency for Food and Drug Administration and Control, also known as NAFDAC. And I don't just want us to recap the content of what was said, but to also reflect on some of my key personal takeaways, because if we're being honest, that session challenged some deeply held assumptions that a lot of us hold, and it also initiated important conversations on what exactly is meant by patient safety. So if I was to begin, one of the key takeaways that I had, and I think Lenias, you can also agree with me, actually came from one of our participants. Her name is Mariam Adenyi, and I quote, she said, "I used to think that a medicine is safe as long as it has a NAFDAC number, and I learned how wrong that assumption can be." Just by show of hands, how many of us have previously assumed that, a medicine that is registered by a regulatory authority is deemed to be safe? You can just thumbs up if that's an assumption that you've held before, that a registered or a regulated product is safe. Okay, so I see that there's thumbs that are coming up. So I think that's really a reflection that a lot of us can relate to. But what we've been taught in terms of looking out for signs, be it registration numbers, labels, packaging, or stamp of approval as proof of safety, needs to be questioned. Because, a medicine passing through a checkpoint of regulatory approval and it getting a marketing authorization does not mean that the job is done. So what we uncovered together and what the last session taught us to question is that medicine safety does not necessarily just work like that, right? So medicine safety is not a one-time check. It is not a non-faltering label, and it's not a guarantee that a medicine will always be safe, because medicine safety is a process that happens on a continuum. And a lot of the times, it's also a fragile process that can be compromised along the different stages of a medicine's life cycle, as well as the different stages that we've got within the supply chain, right? So that was a key takeaway for me personally. But one of the biggest shifts from the last session was also realizing that a medicine's journey does not end at manufacturing. And in fact, it usually begins there, right? Because if we think about it, manufacturing is normally one of the better regulated parts of the convoluted supply chains that we've got. So from the moment that a medicine leaves the manufacturer, it goes through multiple hands and multiple environments. It involves passing through multiple storage facilities, multiple transportation systems, multiple distribution channels, and multiple pharmacies, clinics, and informal markets before it gets to the patient. So at every single one of those points, there is a possibility for something to go wrong. And unfortunately, our lived experience also tells us that there's a lot of things that go wrong. Just by show of hands, how many of us can relate to things going wrong after manufacturing? So I'm sure that there's some of you who work in community pharmacies, some of you work for wholesalers, perhaps distributors. Can you guys relate? You can just put a thumbs up. Okay, I see Yaz is raising her hand. I see David's hand is also raised. So I think we can all relate that every single life cycle point and also every point at the supply chain does create room for medicines to be unsafe, right? So it's important for us to realize, because a medicine can be perfectly made. It can, be GMP compliant and follow, all the ticks, but it can still become unsafe because it was stored at the wrong temperature and humidity conditions. It was transported poorly. It was mishandled by people. It was distributed through unregulated channels. It expired because there was poor forecasting, because there was a lack of transportation, or it was held up at customs. And in some cases, also because it was being prescribed, dispensed, and administered wrongly. So this is important because it changes the question from, was this medicine approved, to what happens to this medicine before and as it reaches the patient. Because like I mentioned earlier on, all of these stages create a loophole that actually leave room for compromised safety. Another important shift for me was expanding on how we define unsafe medicines. I think a lot of the times there's a tendency to equate unsafe medicines with counterfeited medicines. What is you guys' experience in the different settings that you're practicing in? Normally, when we refer to unsafe medicines, does that refer to counterfeited medicine in your setting? If that applies, once again, you can just thumbs up. Okay, I see Yaz is raising her hand up again. I think in a lot of our settings, we equate unsafe medicines with counterfeited or fake drugs, right? But that's usually not the case because unsafe medicines could also be substandard products. They could be falsely labeled products. They could be unregistered medicines, or sometimes even legitimate medicines that have been compromised along the way, as I've stated earlier on, right? So it's important for us to not only focus on the debate of fake versus real, because that actually misses the bigger issue. And we need to focus also on the system that exists behind these medicines, right? And the reason why this is important is that unsafe medicines are not always a result of bad people who are doing bad things. Sometimes they're just the result of weak systems. And when we talk about weak systems, we're talking about unregulated markets. We're talking about poor law enforcement. And I'm happy because Dr. Iliomade's presentation, highlighted the importance of law enforcement as well. It's referring to limited traceability. I remember we had a colleague who also was working on technologies that deal with tracing batches of medicines and working on traceability, and it deals with gaps that we've got in oversight, right? So when all of these systems are weak, risk shows up, and risk doesn't always show up loudly. Sometimes it enters quietly. It becomes normalized, and a lot of the times it can spread and it can compromise safety. And the consequence of this is very important. It's not just theoretical, but it is in practical things that we get to see every day. And some of these things include treatments that don't work, a rise in antimicrobial resistance, patients who keep returning again and again to hospital despite adhering to their course of treatment, complications that come from adverse drug reactions, and in the worst cases, even death, right? So they are very much consequential, and they are things that actually affect patient outcomes and lives. So we're not just talking about products, but we're talking about patient outcomes and lives. And the next point is probably going to make some of us uncomfortable. I recall having this conversation with Lenias as well. Some of us think that we are exempt from unsafe medicines because of where we purchase our medications and where we access our healthcare, right? Just by show of hands, how many of us equate shopping at larger private chain pharmacies and private hospitals with access to safe medicines? If you guys relate, you can just thumbs up, or if that's an assumption that exists widely within your communities, you can just raise your hand up. Because in some of our communities, there is an assumption that going to elite facilities, private sector hospitals, chain pharmacies, that would actually protect us from unsafe medicines, right? So that's also an assumption that's held. But it doesn't account for something that's important, which is, remember, medicines need to be manufactured in safe facilities, safe manufacturers. So even if a medicine is purchased from a trusted source, like your private hospital or your big chain pharmacies, you're not automatically guaranteed to be safe, right? Because sourcing is important. And once there has been a break in one point within the supply chain, that automatically risks everything else. So the end point doesn't necessarily actually protect you from unsafe medicines, right? So it's not just enough to trust the endpoints, but we also have to strengthen the entire system behind it, because if the source is compromised, so is every other point of the supply chain. So this means we need better traceability, stronger accountability, and tighter control across the supply chain. And as we prepare to wrap up, the last point is on awareness, right? And the key takeaway from that session was that awareness matters. And it doesn't just matter, but it matters a lot. Because unsafe medicines don't just circulate because our systems are weak. They also circulate because people don't always know the risks or don't have access to affordable and better options. So whenever we create awareness, we're doing a few things. Awareness helps us to help people actually to make better and safer choices. It helps our professionals to spot the risk earlier on. and it also creates demand for better systems. So in many ways, awareness is where protection begins, and that's why we call it the first line of defense. So unsafe medicines persist only because, not only because of supply chain failures, but because many people also don't know what to look out for. And so by enabling people to actually be aware, we are truly creating the first line of defense. So if I had to actually summarize the last session in one line, it would be that medicine safety is not a one-point check, but it's a continuous responsibility. And yes, a lot of the times regulators are responsible for that responsibility, but it's also up to us as healthcare professionals, as supply chain actors, as policymakers, and as communities. So the reality is that there is a shared public health responsibility, and we all have a role to play in it. So before I wrap up my session, I just want to leave you guys all with 2 questions. And you can answer these questions right now if you're able to come up with the answer quickly, or you might also type it in the chat as we go along with today's session. But the first question is, if medicine safety can break down at so many points within the life cycle and within the supply chain, where do you fit into ensuring patient safety? So I want you guys to have a personal reflection and to tell us, what do you do and where do you fit in terms of ensuring medicine safety? And then the second question is, please just tell us a little bit about what you have been doing up to now in order to ensure patient safety, and if there's anything that you'd like to change following the last webinar and the upcoming ones. Tell us in terms of what you are hoping to do differently after attending the last webinar that we had. So both questions are a little bit reflective, but we want to know in terms of where you fit in within your current role in terms of ensuring patient safety. And secondly, tell us in terms of what you intend to change after the last session that we had. So I think let me not take up too much time. I just want everyone to hold on to these questions and to answer them. You guys can raise your hand if you've got the responses. And without further ado, I just want to say thank you for listening, and I'll hand it back over to Lenias.
Thank you, Lydia. That was an excellent summary of what we talked about. And hopefully it just gives you an idea of what, the conversations we've had in the first webinar, and which we are going to build on today with Dr. Partha Mukherjee, who's just joined us. So welcome, Dr. Partha. I'll just introduce you in a minute, but and then we can start the main webinar session. But the thing to say about this is that addressing the issue of unsafe medicines, whether they're substandard, counterfeited, or other types of, other reasons why medicines become unsafe, it's really something that cannot be solved in isolation by any one group of stakeholders. We need everybody, industry, regulators, communities, the patients themselves, academia, think tanks, all of the above. We need everybody really putting their heads together, because this is a complex and very embedded problem that we really have to find solutions to in order to better protect patients. So what we need is collective cross-sectoral leadership that is bringing together industry, regulators, clinicians, pharmacists, and all of the frontline healthcare professionals, in addition to the one that I just mentioned. And this flagship webinar series is really designed to treat unsafe medicines as a practical, ongoing, and urgent safety challenge that has not so far been on the agenda of African countries. And we want to bring it on the agenda, and we want to catalyze action in order to protect patients safer. And so the webinar series really aims to equip stakeholders with actionable tools to identify risks early and to reduce harm in real time. And this is what Dr. Partha is going to help us today. I think he's going to make some recommendations for supply chain actors, recommendations for national regulatory authorities, for marketing authorization holders, and everybody. I think we have something to take away of things and tools that we can be able to look at this challenge in order to from growing from awareness to prevention, detection, and taking real action. So and what makes this webinar series different, right? What's different about it is it's grounding in frontline learned insights with real world case studies for from the African context. So far, we have case studies that are coming from 8 countries and growing, because we this is very early days. We have more webinars coming, so I encourage you to tune in, because there's so much that we will be able to learn from people who are really involved in taking action against this. And so, and today, I guess, for the benefit of the speaker in the room, we have people who are joining from NRAs, regulatory authorities, from ZAMRA, SAPRA, EDA, and many others, BOMRA, there are many of them joining tonight. And also from big pharma companies like Bayer, Novartis, GSK, Eli Lilly. There's even Unilever. Didn't know they were involved in this space, but here we go. Welcome. We also have participants from generic manufacturers like Stride Shasum and others, Energia Pharma. So it's really a great mix of people who have a very specific interest in this. Of course, this is in addition to our frontline healthcare professionals, our pharmacists and clinicians who are joining. We have some from Wits University and universities from Nigeria, Zambia, from Europe, Italy, Belgium. So this is the kind of audience you're going to be speaking to. And so, Dr. Partha, just to recap, in our previous session, we explored the scale and agency of unsafe medicines with Dr. Martins Iliomade, who is the Director of Investigations and Enforcement at NAFDAC. And he reminded us that the problem of unsafe medicines is no longer a hidden issue. It's a full-blown crisis that needs urgent access. Sorry, there were some voices there. So today we are going to build on that foundation by making a very critical distinction that not all unsafe medicines are the same. And treating all risks as identical can actually lead to an ineffective response. So we need to differentiate them in order to have the right response to the right risk. And so today we are going to deepen our focus on degraded medicines and why clear differentiation between the categories of unsafe medicines matters in practice. And so, distinguishing these categories really is going to equip frontline healthcare professionals to be better equipped to detect risks earlier, to respond appropriately, and to ultimately be more effective at protecting patients. And so without further ado, it is my pleasure to introduce our speaker today, who's going to take us through today's webinar, Dr. Partha Mukherjee, who is an executive director for analytical chemistry at Eli Lilly and Company. Welcome, sir. I hope you will be able to really learn a lot from the case study that he brings today. But Dr. Partha brings very extensive global leadership experience in analytical pharmaceutical development. He has held senior leadership roles at 5 of the top 10 global pharma companies. So there's quite a lot of institutional knowledge, in his head. And his expertise spans the full product life cycle from preclinical, early development through late stage development of products and commercial manufacturing support. He has led global initiatives in R&D, and commercial stability programs, as well as reference standard systems. I believe he's worked with USP and, helping to ensure the quality and consistency of medicines at scale. So he is widely recognized for building and strengthening analytical centers of excellence. So hopefully we will learn a little bit about that in our African context, where all of this is still very nascent, and we are still trying to build up that knowledge base to know how to deal with some of these challenges that we haven't always tackled. And so we are honored to have you, Dr. Partha.
Thank you.
Today to share your insights on medicine's quality, analytical science, and the emerging challenge around nitrosamines in pharmaceuticals. So I pass it on to you. Welcome to you. It should be good morning where you are. I think good evening here.
Oh yeah, it's 12:30. So yeah, you can say good afternoon. Yeah, that's and Lenias, I'm wondering whether it will be better for me to drive the slides, or you want to do that? I think just to give you a heads up, the internet bandwidth sometimes is a little bit, faulty. So if you see things getting stuck for a couple of seconds, don't bother. It will come back again. Thank you, first of all, to Medicines for Africa and Lenias for giving this great opportunity to bring a very hot topic, very hot industry topic to the African continent. And I'm really honored to have this opportunity to share some of my experience, real hands-on experience on nitrosamine. To begin with, I should say it's basically a global problem. Africa has its own problem. It may be a little bit more compared to EU and US and other countries. However, nitrosamine is a red flag for any country globally, and there is still the issue happening in terms of properly tackling nitrosamine in new medications. So my goal today is to give you a case study example. And as Lenius mentioned, this is a multi-prong approach. So not only there will be elements of chemistry, because nitrosamine will heavily depend on the chemistry, structural chemistry. There is the analytical chemistry. There is also formulation, regulatory aspects. There is, all aspects are there. Toxicology, of course, and then clinical, activities, etc. So it's a multifunctional theme for controlling nitrosamine. So I would probably give you a very good idea on how to take stepwise through the nitrosamine challenge and how to mitigate that. So that could be used as a guidance. I have got proven fact that the case study that I will take you through, that has been very useful for various other communities, scientific communities worldwide. I have presented that in many platforms before. I'll touch upon also the nitrosamine landscape in Africa based on my knowledge and based on what's the information out there, based on various surveys, etc., and give some recommendation. That would be my goal. So just to begin with, we have to little bit delve into chemistry, as I mentioned, because otherwise you won't know how the nitrosamines are formed. I have to give you some background on that. So nitrosamines basically is the structure that you can see on the screen. It's N-nitroso. That's the kind of trigger point. So NNO, nitrogen, nitrogen, oxygen. That's the structure. And then you have R1 and R2. These are the two different functional groups attached to it. So wherever in the structure in a new compound, if you see these structure showing up, that's an indicator that, oh, we are dealing with a nitrosamine. So this is a control strategy that I will take you through, and this is part of a group called Cohort of Concern, or COC, as part of the ICH-M7 guideline. Hopefully all of you are familiar with that. It's the International Consortium on Quality and Harmonization. They have a guideline on mutagenic impurities. And these are considered, the nitrosamine is considered a highly potent mutagenic carcinogen in many animal species. And that's why there is red flag on this when it comes to humans also. So look for that structure. That's the first, thing to remember. There are two separate types of, mainly two broad categories of nitrosamines. One is the standard nitrosamine, which is on your right side. You can see 6 or 8 different structures: NDMA, NDEA. These are the abbreviated names. I won't go through those, but most of the regulatory bodies in the global community, they are familiar with these names because that's how the nitrosamine journey started back in 2018 when it was first found in valsartan. NDMA was found, and that triggered the recall, and then it started to expand to various other therapeutic areas, like blood pressure medications, obesity, and then the proton pump inhibitors, those kind of thing, or the gastric, the different components, and diabetes drugs like sitagliptin, etc. So that's one category, the standard small nitrosamine. And then the one which is more recent, I would say, which started in 2021, is the NDSRI. These are the most dangerous compounds because they are very specific to API. You cannot standardize the things which you can do for standard nitrosamines. You have the regulatory authorities, they have set up the acceptable intake limits for these, so it's a little bit easier to control in a way. The NDSRIs, on the other hand, they have their own acceptable intake limit that's fully based on the structure of the active pharmaceutical ingredient. That journey started in 2021 when there was a NDSRI found with this compound Chantix, which is a smoking cessation product. And the latest one, just last month actually, there was one which was reported for NDSRI recall. So this has gained tremendous visibility for global regulatory bodies, and they are focusing and scrutinizing on each new medicine that's coming out, and can they form any NDSRI. So my talk will be more focusing on the NDSRI part, because the standard nitrosamine, there are tons of methods out there for controlling those from different regulatory bodies like FDA and EMA. So that journey started in 2021 when there was a NDSRI found with this compound Chantix, which is a smoking cessation product. And the latest one, just last month actually, there was one which was reported for NDSRI recall. So this has gained tremendous visibility for global regulatory bodies, and they are focusing and scrutinizing on each new medicine that's coming out, and can they form any NDSRI. So my talk will be more focusing on the NDSRI part, because the standard nitrosamine, there are tons of methods out there for controlling those from different regulatory bodies like FDA and EMA. So now, slowly getting in the nitrosamine overall scenario in Africa, there are three main sources where from the nitrosamine can come, as has been reported in literature. One is the drinking water, groundwater and surface water. The other one is the diet and traditional foods. So this is interesting because there are some foods which can endogenously form nitrosamine, and that is now gaining a lot of attention from all the regulatory bodies, like especially I would say like FDA, EMA. They are looking in all these also, that food intake obviously can create a lot of more nitrosamine than the exogenous source of nitrosamine, which comes from the pharmaceutical products. The population is going to be, obviously the vulnerable ones is infants, young children, elderly populations, pregnant women, etc. And my focus obviously is going to be on the pharmaceutical products. That's where the nitrosamine I'm going to focus in my talk. So in medicines, just an overview, there are several sources from which the nitrosamine can come in the drug product or finished pharmaceutical product, as we call it. It can come from the excipients, it can come from the APIs, and you see all these things. Utilities, manufacturing process, primary packaging, lot of various factors to control when you're thinking of the overall risk mitigation for nitrosamine. So the package that goes in the dossier submission for global regulatory bodies typically is a very comprehensive one, where you have to show that all these different criteria which are listed here, they are being controlled, they are being identified, and they are being controlled. And that's the main, aspect of this. uh, and then I want to show also the rapid evolution of nitrosamines. It started with small nitrosamine like NDMA, as I said, 2018. But look how it has been exponentially growing. 2021, the first NDSRI showed up in nitrosovarenin clean. And then now, as of today, these are the things which are mentioned in this, blue box. There are structurally more complex NDSRIs discovered. In one drug product, there could be multiple NDSRIs forming, which I will show in my example actually. More stringent health authority controls, new ways of AI calculation. Some are, proposing quantum mechanics over the latest approach, which is CPCA, and I will go in that little bit more details. Endogenous nitrosamine formation. There are a lot of literature reports coming in false positive nitrosamine when really there is no nitrosamine, but it's triggering a false alarm. Nitrosamine can form by air exposure of APIs, which is very, very dangerous thing, right? Complexity of the formulation. Solutions can create more NDSRIs compared to solid. So oral solution will have more nitrosamine compared to a tablet or capsule for the same API. Use of AI, artificial intelligence in nitrosamine control, that's getting a lot of prominence. Automated nitrosamine analyzer, this is a huge thing happening to increase the efficiency of productivity of identifying, rapidly screening the medicines, how you can get the total nitrosamine picture in a molecule. And then predictive stability, while it's in the storage, how much nitrosamine can build up. So there are a lot of predictive stability happening. So each of these can be a huge seminar, individual seminar topics. So we'll touch upon that later. And just to share, before I deep dive in the case study, there are a lot of resources out there. I am personally very proponent of this USP's nitrosamine exchange platform, which is more than 5,000 members right now. They have such resource on nitrosamine, available in this particular exchange community. So I really encourage you guys to look into that. They are being developing a risk assessment practical tool for nitrosamine monitoring, and that's also I was involved in this tool actively. That's going to be very useful tool. And then there are a lot of other sources. If you go in the websites of individual major regulatory bodies, they have a lot of analytical methods and details. So that's out there. So now coming to the case study for risk assessment and life cycle management of NDSRI. So my goal will be by end of this talk, you will have a good idea how, without knowing anything on nitrosamine, if you have to control and if you have to propose something to regulatory bodies, what are the steps that you need to follow? Okay, and this is based on my personal successful experience with EMA. So the way it started is we had a compound miglustat, and that we wanted to get it approved in EMA. That's where they came back and said, have you done any nitrosamine assessment, like specifically on the NDSRI assessment for the nitrosamine in miglustat? And it had an process impurity, L-idomiglustat. These are all amine compounds. And is there possibility for formation of NDSRI by reaction with the nitrite of the excipients that you are using in the formulation? If so, share us the information and what's your testing plan. At that time, we didn't have much insight on this thing. The consultant that we had borrowed, hired basically didn't have a very good in-depth knowledge on this thing. So what we proposed, EMA came back and said, nope, this is not acceptable in terms of the risk assessment. This will be a major objection. So major objection means that's a red flag right there. They're not going to approve until you come back with the proper risk benefit profile and all the risk mitigation resolution, before the marketing authorization can be granted. So it's putting the drug on hold, basically, the approval. So implication was very, very prominent and very deep implications, I would say, because if this drug didn't get approved and EMA doesn't approve it, that has a cascading effect, because EMA has the most stringent nitrosamine regulations of all the global authorities, as I know. If we cannot pass that, then we cannot pass other countries like FDA, MHRA for UK, TGA for Australia, PMDA for Japan, Swissmedic, all the other countries, it will be on hold. So, and as I said, like, we didn't have the internal knowledge and the knowledge for all the consultant industry CDMOs. They were also evolving. So I was actually asked to take this on because I had some previous knowledge, and that's how my journey began for this one. So very high level summary, these are the things that you have to consider. So very important facts here. A comprehensive life cycle management approach for NARA, that is nitrosamine risk assessment, needs to have these 6 steps. You start with a chemistry risk assessment, left to right I'm moving, that needs to show that can your compound form a nitrosamine, specifically NDSRI. And I'm talking with NDSRI because that's the most complex nitrosamine we have to deal with. So once you show the chemistry risk assessment, then there is the tox risk assessment. That will have to show that what is the mutagenicity and carcinogenicity potential of your NDSRIs, the proposed NDSRIs. And then you have to come up with the acceptable intake limit. And there are various ways to do that. Then you come up with the analytical risk assessment, which means, okay, we know that how much is the acceptable intake limit for this compound. Now we need a method to monitor that, and that's where the ERA kicks in. And basically that needs to be very sensitive method. It needs to be the LOQ, or limit of quantitation, is not more than 10% of the acceptable intake limit that you have established with your tox risk assessment. Okay, so once that is done, then you have the in vivo metabolic bioactivation RA, or risk assessment. That means, okay, we know that maybe 5 ppm is active, daily basis, it's getting ingested through the tablet or capsule, maximum daily dose. However, there is still a possibility that they may not get metabolically activated inside the body. And that's where you can actually reduce or downgrade your risk for NDSRI, by showing that in reality, metabolically, this will get eliminated. The risk is further reduced. And then you have the clinical risk assessment that's based on the dosage form, whether it's oral solution versus tablet, capsule. What is the maximum daily dose? What is the frequency of dosing? What is the target population, etc.? Everything needs to be mentioned. And then comes the final one, which is before your dossier submission, that is your regulatory risk assessment. The probability of success, or POA, that how much confident you are with your package in terms of getting it through the IND IMPD stage. So the thing to note is the regulatory bodies are starting to ask for nitrosamine risk assessment way early in the process when you're filing an IND or IMPD. Before it was in the final package like NDA or MAA, but now the process has moved way up in the submission, and they are starting to ask for this information during IND-IMPD itself. So little bit of chemistry, I'll keep it very high level. For secondary amines and tertiary amines, these are the most vulnerable moieties for APIs that form nitrosamine. So the key thing is you have the nitrite, which is actually coming from the excipients that forms a nitrous acid, and that nitrous acid again breaks down and that forms the NNO. This is the moiety which actually is the activating species, and that reacts with a secondary amine, and that forms the nitrosamine. For tertiary amine, which is another step is involved, and that's where the nitrite actually goes through a more elongated step, and then it forms again nitrosamine, but it goes through a secondary amine step. So the key thing is the reaction of secondary amine to form a nitrosamine is 1 to 1. For tertiary amine, the reaction is you need two molecules of nitrite from the excipient to react with one molecule of your compound, and that forms one molecule of your nitrosamine. So this is the key thing to remember, the reaction. That's the basic. Okay. For our miglustat compound, I'm not going through the details, but this is the structure. This has the tertiary structure, tertiary amine, that undergoes the steps as I mentioned. For tertiary amine, there are long steps and two molecules of nitrite, finally giving the N-nitroso-miglustat. So the nomenclature for NDSRI is N-nitroso and then the name of the API. That's the common thing, right? So here the name is N-nitroso-miglustat. For its process impurity, which is also a tertiary amine, L-idomiglustat, that undergoes the similar conversion, and that forms N-nitroso L-idomiglustat. Okay. Look at the structures; they are almost identical, and I will show that, and that's what makes the process, the project more challenging. By the way, these excipients came from microcrystalline cellulose and starch. Those are the two excipients which contributed the nitrite that reacted with, potentially can react with your API and the impurity. So once you say that, okay, there is nitrite in excipients, you have determined that from your vendor certificate of analysis. The next step, what regulatory bodies require is show me how much nitrite per capsule, per tablet, or per dosage form oral solution you have. So you have to do this calculation where you show that how much actually nitrite is coming from the excipients total per capsule or per tablet, and then you do the math, and then it shows that how much of the total NDSRI can form. In our case, the NDSRI per 65 milligram capsule of miglustat, that turned out to be 2 microgram of NDSRI, and that is the combined amount from the two separate NDSRIs, one from N-nitrosomiglustat, the other one is N-nitroso-L-idomiglustat. And then after that there is something you have to take into consideration, that okay, we know theoretically this much can form, but in reality how much will be there? So then there is something called a Scriven's model, and then there is also microenvironment pH. The slides will be shared hopefully, so all the details will be there. I don't have just the time to go through all the details, but these are called the reduction factor. So in general, what it means is, if you calculate, let's say you have 10 milligram of nitrite in a capsule coming from the excipients total, in reality all of that nitrite is not actually available to react with your API or whatever the amine in that capsule. There is a reduction factor which can be 10% or maximum of 30% of the available nitrite actually reacts to form the NDSRI. So that's one reduction factor. The other one is called microenvironment pH, which means basically the reaction rate gets much less compared to the theoretical prediction, because you are dealing with, let's say, solid dose, where the reaction kinetics is almost thousandfold lower compared to in solution, oral solution. So considering all those, there is also the fact that the practical aspects, the tertiary amines typically react almost thousandfold lower than a secondary amine in solid state. Then there is something called steric hindrance. Means if you remember the structure I showed, where that's nitros, the NH group is there, the amine structure. If there are bulky functional groups around that, then that prevents the reaction between nitrite and your NH or the amino group. So that's another way of reduction in the chemistry. And then the actual manufacturing process, the dry granulation obviously will have less reactivity because there is no water. Water is a culprit for triggering a lot of these reactions. However, so we showed that this is what we presented to regulatory bodies, that in reality there is the very little risk of formation of nitrosamine. But since we said that there is potential for formation, then we had to take the next step, which is the toxicity risk assessment and the acceptable intake establishment. um, this is just a slide just to show that there are different ways this nitrite level can be calculated for the risk. Something called FMEA, or failure mode effect analysis. This is something industry is starting to do recently to show the chemistry risk assessment part for synthesis of these nitrosamines. Um, so we know that on paper we have shown that there are two nitrosamine that can form, and then we have to use a in silico toxicology risk assessment tool, which is direct nexus and Sera nexus. So the direct nexus is used for predicting mutagenicity of your NDSRI, and Sera nexus, this predicts the carcinogenicity. So these two actually gives you the data. You have to input the structure in this software, and then they spit out basically some numbers. The statistical probability of mutagenicity and carcinogenicity is this much. So once you know that, then you have to actually come up with the acceptable intake limit for those NDSRIs. That, okay, your software in silico risk assessment showed that there is plausible mutagenicity and carcinogenicity. How you're going to control those? And right now there are different ways to do that. When I was doing this project, there were two options available. One is the TTC, threshold of toxicological concern, and that has a default limit of 18 nanograms per day. The other one is based on the structure activity relation, or SAR, and that one is basically I will show that one in the next few slides. That's another approach which is approved by EMA and all the regulatory bodies. Now, currently, for the last year or so, the more used method is called CPCA, or carcinogenic potency categorization approach. That's based on just the structure of your NDSRI. You can actually predict what would be your potency category in terms of carcinogenicity and all those things. So I actually, in this case, use the SAR approach. And if you look at the extreme two right structures, those are the structures of the two NDSRI. One is the N-nitrosomiglustat, the other one is N-nitroso-L-idomiglustat. These two structures were input in the software, and the structural similarity is with N-nitrosopiperidine and two other compounds. This is called a structural surrogate approach. Means these compounds that extreme, these three compounds in the green box, they have been already well characterized in terms of their toxicity risk assessment. They have a TD50 value, toxicity dose 50, that lethal dose value, And based on structural similarity, these can be used as surrogate to adopt these, values for, evaluation of these on a paper based, because we have no idea on the toxicity of these, newly synthesized NDSRI. So N-nitrosypiperidine, that has a acceptable intake limit of 1300 nanogram per day. And then this one was chosen because this is the most potent we found in terms of the TD50 value. So we wanted to be in the most conservative approach to show to EMA that, hey, look, we are using the most conservative approach when we are establishing our acceptable intake limit for these two NDSRIs. They are structurally similar, and based on that, then we can adopt probably 1300 nanogram per day for our two NDSRIs. Okay. But before that, we have to get the buy-in from EMA, and that's what I had the meeting with the scientific advisory working group. Do they agree with this approach, and do they agree with the surrogate that we are choosing in terms of our, acceptable intake evaluation? They said, yeah, it's fine. However, they also cautioned us that the actual decision that how much your acceptable intake limit will be for a new nitrosamine, that's being determined by NCWP, this group. Okay. So without toxicity data, we should be more cautious and use the most conservative approach of 18 nanograms per day, which means basically they are suggesting that yes, it's fine to use SAR, but since you don't have any data, go with the most conservative TTC approach, which is 18 nanograms per day. Okay. So that's the acceptable intake. And then there is the calculation that you have to be aware. Anyone working in nitrosamine needs to be aware how to convert the acceptable intake of a nitrosamine to the acceptable intake limit. So all you do is just divide your AI by maximum daily dose. In our case, it was 4 capsules of 65 milligram each. And that turns out to be 69 parts per billion in the NDSRI. And then what would be your target method sensitivity for the analytical method? That has to be 10% of your AI limit. And that brings down to 6.9 parts per billion. Can you imagine the sensitivity that you're looking for? And that is just an example how sensitive for NDSRIs the method needs to be. So that's our method development target. CPCA approach, as I mentioned briefly, we did also the calculation after we submitted the dossier. And it turns out, based on the structure, the different features, it turns out the EI is 1500 nanograms per day. So here is the picture. So CPCA approach, which is the latest approach, if we had used that, our NDSRI acceptable intake would be 1500 nanograms per day. We used SAR approach, which is structural surrogate approach, that gave us 1300 nanograms per day. But we still went with the most conservative approach, which is the TTC approach, toxicological threshold of toxicological concern, that is 18 nanograms per day. If we had done the method development today, probably we would have gone with CPCA and not all this, complex challenges with method development by TTC approach. In the interest of time, I'm going to skip this one. The main thing to show here is that the APIs that you get from multiple vendors, let's say you have the sourcing from two different vendors, they may have different impurity profile for amines. That means your NDSRI profile could be different based on which API you are going to use. That's a very important thing. The other thing is we really don't have a very good idea when the NDSRI formation happens, because there are so many contributing factors. It's pH, could be water content, temperature, pressure, the catalyst available. So it can get really, really very complicated. That's what I'm, trying to say here. In our case, when we started on the method development for analytical, just look at the structures. They are almost identical. The only difference is where you have this, the CH2OH, that methylene hydroxy group adjacent to the NNO, or the nitrosamine, moiety. That's where one is the hydroxyl group is sticking out from the plane of your molecule, and the other one is it's behind the plane of your molecule. So just a stereochemistry difference, that's it. Everything else is identical. So in terms of analytical method development, this is a major challenge, and that's what we actually had to deal with. The target quantitation limit was also very, very low, 6.9 ppb. You are looking at extremely sensitivity. And then there are a lot of excipients also, so they can create a lot of interference in your analytical method. Okay, so a lot of challenges. And then what we did is we put together three different CDMOs, the resource, total of 16 chromatography, which is the separation, science, subject matter experts, and we tried lot of different conditions. I'm keeping it again very high level. 24 different columns which are listed here, and 29 separation conditions were employed by 16 different analytical subject matter experts because of the complexity of this. And we had the clock ticking. EMA said you have 3 months of time to get this, your response back. I'm going to skip this. This is for more for folks who have, you are in the analytical folks or the QC folks, just to show that the final condition that we got, in terms of the separation of the two NDSRIs, we actually could not separate out these two NDSRIs because despite all the challenges, this is the best condition we could get. And then this is the, showing the peak that we show, here at 3.19 minutes. It's a using LCMS. So that's another critical thing for nitrosamine analysis. The very basic analytical instrumentation that you need is a LCMS, liquid chromatography mass spectrometry. That's the basic thing. Without that, you cannot do any nitrosamine analysis. It's an expensive instrument. It requires a lot of subject matter, expertise, and it requires a lot of care, and maintenance, etc. So we couldn't separate it out, but we actually could convince EMA that we have tried all reasonable things. And typically the regulatory bodies, they're very reasonable on this thing. So that's how it went. And we came up with one peak for combined nitrosamine, which is also a very unique approach in regulatory field. But we did that. And then we validated the method, again not going through the details, but these are the typical parameters that you have to validate: the specificity, detection limit, linearity, accuracy, repeatability, etc. All of those are based on ICH guideline, ICH-Q2 guideline for method validation, and we showed that our method is very robust. Okay, after that, you have to use the validated method for confirmatory testing, and you choose three different batches of drug product. Preferably, they are made from three different API, using three different API batches. And we showed that all these are below the quantitation limit. So once you can show that, then you do not have to test for the nitrosamines in any release, batches. So that's the great thing. And then finally, you have to pull together the NDSRI risk, assessment in one tabular form, which we did here in terms of chemistry, tox risk assessment, clinical risk assessment. We showed that the NDSRI risk level is negligible for all of those. This is something kind of it will be great to have in a regulatory package, just to highlight the summary of the whole activities. Okay. And then the life cycle management is basically three stages. You have the stage one, which is your method development, knowledge gathering. Stage two is you actually do the process, qualification. which is basically validation, and then the ongoing procedure performance verification, that's the stage 3, which is the confirmatory testing. So this is expectation from regulatory, that you have to show the life cycle management and you have good control strategy on this thing. Okay. So novelty of this project was basically we came up with 18 nanograms per day, and we were able to fulfill that. At the same time, the method was so sensitive, it actually also fulfilled the other ways of calculation of NDSRI intake limit, whether it was SAR or CPCA. We did that, and this was the first report actually where we encompassed all the risk assessment stages in one risk assessment document. So this was actually used, EMA just agreed and approved it in one shot after once this is presented. And this was presented, it's actually published and being used as a guide for a lot of the companies and regulatory bodies in terms of how a typical risk assessment should look like, especially for NDSRIs. Okay. So I have basically my recipe for success, stepwise guidance. You can go through these. As I said, like first start with whether your API has secondary, tertiary amine. Then look at the excipients. Do they have nitrites? If they have, then do a paper-based chemistry risk assessment. Find out what is the structure of your NDSRIs. If you find that out, can you synthesize that? That will be the next step. If you can synthesize that, then you have to go through the steps for 4 through 11. If you cannot show, you cannot synthesize, you have to show that to regulatory bodies that we have tried all reasonable efforts. On paper, yes, they are there, but in reality we cannot synthesize that. So that is a rationale for justification that you don't have to go through the actual method development and things like that. Okay, so then calculate the AI limit. As I said, they're using CPCA or SAR approach, come up with the proper analytical method, validate the method, and use that method for confirmatory testing of your batches. If it turns out that your limit is more than 10%, then you have to establish your specification in the drug product control strategy, and that's where it can be based on the most potent NDSRI AI, or there is a flexible AI approach. And these are very well outlined in US FDA or EMA nitrosamine guidelines. You can follow those. So yeah, these are the key things. So this is kind of the takeaway, single slide. What are the steps that should be followed, to doing the NDSRI lifecycle management or risk mitigation? Coming to now the more relevant, I guess, the African portion of the nitrosamine. So in terms of the landscape, as you know, and probably has been already described in this webinar, and all of you should be familiar, Africa continent actually imports 70 to 80% of its medicines, and that makes it a lot of reliability on the supply chain, the global supply chains. It imports over 95% of the APIs, mainly from India and China, and so the production becomes expensive, vulnerable. However, a lot of the, several of the African pharmaceutical companies, they are now taking over and trying to come up with these, manufacture of APIs locally. There are a lot of investment also happening. It looks like it will require a significant investment, almost like $11 billion, based on a paper report actually, that is required to make a little bit more self-reliance on the API manufacturing side. So knowing that 70 to 80% of the imported medicines are coming in Africa, what are the NDSRI or nitrosamine challenges? So these are the main ones. You have the regulatory challenge. There are three interconnected challenges that I could think of. One is limited analytical capacity or capability. So you will see the theme. I will be repeating it probably few times throughout the remainder of the talk, but I want to drive these points that building the analytical capacity is going to be a key issue for, key, I would say, area for the African continent. Detection of nitrosamine requires very advanced analytical methods, high-end instrumentation in terms of cost, as I said, the maintenance, the running of the instruments, etc. Those needs to be there. Um, Also, the work sharing initiative, which is happening through Zazibona, which is pretty good. However, the communication with the marketing authorization holders can be slow due to limited, regulatory capacity, and there are some survey output I can share on that. Um, then the risk assessment and compliance. So again, NDSRI, this is going to be the most challenging portion of the whole nitrosamine journey for Africa in going forward. And the reason is basically NDSRIs are API specific, and they can be very complicated molecules and very difficult to monitor in terms of their analytical sensitivity requirements. um, Operational and supply changes, the testing and confirmatory analysis for the imported medicines can delay product registration, creating medicines shortage. Okay, so these are the challenges with the imported medicines. And then on top of that, you have the African climate. So very extreme climate. It's a zone 4B, which is, basically 30-degree and 75, or 65 and higher relative humidity. And those are the ideal platform for more NDSRI formation. So that's the additional complexity of drugs which are getting manufactured in Africa or coming imported in Africa. During their shelf life, they can have these challenges because of the climate. High temperature, high humidity, hygroscopic excipients, non-optimal packaging means it was probably meant for, temperate climate, the packaging, and that is not able to sustain the heat and moisture transfer. So impurities will grow. Storage duration, how it was stored in the right condition or not. So a lot of the patient education that needs to be also done. So these are the key factors when it comes to the challenge on that. Now, I did a little bit of search, and looks like there are various countries within Africa which have some sort of nitrosamine guidelines. South Africa is obviously SAFRA leading on that, and there are these other countries listed which have some regulations listed to various extent to address the presence of nitrosamine regulations. The typical medicines which have been identified with nitrosamine issues, I'm sure all of you are aware. There is the beta blockers, then the antibiotics, diabetes drugs, GI drugs, smoking cessation, etc. Um, so there is the nice regulatory approach already there in terms of standard nitrosamines, as I said, risk assessment, confirmatory testing, reporting, and control. What I find a little bit as a kind of gap area or area to, I'd say, where it can be better is the NDSRI life cycle management. As I mentioned, that's more complex nitrosamines. Zazibona, the collaborative, initiative, they are playing a big role. What I'm aware of, they have several countries, listed in that, and basically they are serving as a model for low and middle income countries in pooling regulatory capacities and also efficient assessment through harmonized guidelines. And there are a couple of templates for data submission, regular training, stakeholder engagement, etc. So the testing for nitrosamine risks via coordinated regulatory assessment, and then these different kind of advanced instrumentation is required, adherence to internationally recommended impurity limits, and so on. So I think it's on the right track. And this collaboration is doing a great job actually in terms of spreading the awareness and trying to streamline the process across the different countries. Very busy slide, but I just want to highlight a very important survey output that came out from this in terms of the experience of the national regulatory agencies in Africa. They're saying most labs do not have access to the testing methods, etc. Need a further regional collaboration. Large number of applications is sometimes overwhelming the NRAs. Then, in terms of having accredited laboratories, that probably is a gap area. Advocacy webinars can help. These are all the positives and gaps that have been identified. Advocacy webinars could be helpful. Collaborative reliance, as I mentioned, that will be helpful. And share the key lessons after action review. That is going to be very important when you actually go through the process, like the case study I showed. It will be great to highlight what could have been done better in terms of the sharing the knowledge with the greater community. And that will actually help to build more trust in the regulatory processes that will be promoted. Improvement in compliance in the administration of medicines by consumers within the region. Very busy slide, but some very key points mentioned here. So coming to my recommendations, I will try to go through this quickly. Again, if you have time later on, you will be able to go through those in details, and we can catch offline. So for marketing authorization holders and NRS, build more awareness on what a robust NARA should look like. The example I gave: continue routine collaboration and knowledge management. This is very important because it's evolving so fast. I cannot keep up with the global regulatory nitrosamine guidelines. There is almost every month one or the other regulatory bodies, they're updating their regulatory guidelines. So the competitive regulatory intelligence monitoring, that will be very important for NRS. Bring the strong interpretation of the guidelines out there. The pharmacopoeial guidelines are also very important. Building a central database could be actually helpful. So one-stop shop. This is something like the Nitrosamine Center of Excellence, which I created for my previous companies. This has the one area where all the product, their nitrosamine information is consolidated. What are the methods out there? What is the regulatory standing of those individual products with the different global regulatory bodies? That kind of central database, or I would say, build a nitrosamine center of excellence for the different regions, which will have the right analytical instrumentation, the right expertise. They will be sharing through webinars their information with the regulatory bodies, the MAH, etc. Then I have some recommendation. Again, a lot of information. I have put a lot of good thoughts, so take your time to go through those. For pharmacists and clinicians, be aware of the recall, drug quality defect, shortage, all through on the different compounds based on nitrosamine. There are some nice links I have included here. Then for the clinician or healthcare practitioners, patient safety and risk assessment. So this is now up to them to make sure that if a drug recall is happening, what is the alternate or contingency planning to put the patient in the alternate equivalent medicine, for example, right? So those are the things which will be very important to do the patient safety and risk assessment. Communicating with the patients in terms of the risk benefit balance, availability of alternates, monitoring drug supply based on the voluntary recalls or mandatory recalls that comes out based on nitrosamines. Collaboration, it needs to be, as as Lydia and Lenias mentioned, basically this needs to be a multi-concerted effort. It's just not individual, just regulatory bodies. No, It has to be down to the patient level. So manufacturers, they start their primary responsibility, but then regulatory bodies, the clinicians, pharmacists, the patients, everyone will have to play a role in terms of how to control this holistically. So that's very important message. Supply chain, I think this was mentioned just before I jumped in my talk. Lot of factors that you have to take into account: the raw material supplier control, how robust is your due diligence for selecting a supplier, are you doing the right auditing in terms of their process, traceability of the batch records, then supply chain storage, are they taking into account the environmental controls during transportation, short term excursion, etc. So there are a lot of things which again is here, the continuous improvement and training, which will be very important. It requires a holistic supply chain strategy spanning from raw material control, process optimization, analytical testing, supplier qualification, and compliance with global regulatory bodies. So keep this in mind, and this is again a very intricate problem, but it can be fixed. So my 6 point recommendation, kind of trying to wrap up in next few minutes: strengthen the local lab capacity. As I said, this is very important. Adopt reliance frameworks from very established, regulatory bodies out there. Standardize the testing protocols. So if you are adopting CPCA, make it consistent across all the countries. CPCA will be the way to go. If there is exception, some MAH, they are asking for SAR approach. Yes, that's fine with proper justification. Implement continuous monitoring with the periodic batch testing for short-term medicines. Um, Stakeholder engagement, promote that across NRAs, manufacturer, healthcare providers. Dealing with climate, so this is something many people forget, the effect of the climate on NDSRI. That needs to be also kept in mind. Okay, I have presented a case study which gives a hopefully good idea on how it should be done, right from the scratch paper based exercise all the way to actual lab testing of the batches and the life cycle management of that. And then I have provided recommendations which are for the MAH, NRS, clinicians, pharmacists, and supply chain professionals. Take a look at that. Those are based on very practical experience and interaction with all these different stakeholders. Holistic nitrosamine control, it needs to go beyond just medicines. Medicine, if you think about it, you're just having a small portion of that getting through the medicine. The bigger picture is the water and food and everything. So that's pretty much it. So, and again, I'll be happy to partner as applicable as the time permits, in tackling this critical safety aspect for medicines. So that's pretty much it. And I have just I'll hand it back to Lenias. It's a big topic, and I tried my best to cover and give you some flavor on this thing. Okay.
Thank you very much, Partha. That was a real tour de force. Very, very informative. And I mean, clearly, this is a big challenge that we haven't even begun to look at in the African context. And that's the purpose of this webinar, to sensitize our community of healthcare professionals, of marketing authorization holders, of national regulatory authorities, and of course the community of our youths as well, who are here, who are involved in all of this. And I hope that for the young people in the room, I hope all the chemistry didn't scare you. I think the take-home message here is that this is a problem.
Yes.
It's affecting, commonly used medicines such as antibiotics, beta blockers, anti-diabetics. And so it's obviously an issue for our communities, for our patients, and something that we need to be doing something about. So and thank you very much, for the audience for hanging on. I know on this side of the planet, it's dinner time. We are all hungry,
Absolutely.
so
Appreciate that. Yeah,
so
appreciate the
great.
hanging for that. Yeah.
Yes. So our audience have some questions, Partha, and I think we'll take them in batches. Can I encourage everyone to introduce yourself first, and then state your question as briefly as you can, because there are quite a few questions and we are already over time, and we'll do them in batches so that Partha can answer. Partha, can I just ask you to stop sharing your screen?
Yeah,
So
absolutely.
we can get the questions going. So I have one participant here, Lakson Goma. Please put on your camera, introduce yourself, your name and your affiliation, and ask your question. Over to you, Lakson.
Are you able to see me?
Not yet.
No.
But we can hear you, so it's okay.
Okay.
I mean,
Yes.
you can go ahead.
Yeah, I'm a registration officer under human medicine. I'm working for the Zambia Medicines Regulatory Authority as an assessor for human medicines. And quite interesting topic that you have presented on. And
Thank you.
Well, okay. So my question, getting direct to the question, is with regards to nitrosamines, are we required to have the same control limits? Okay. Yes. So do we have to control the nitrosamines and have the same approach regardless of the duration of treatment? I understand. That does the model that we're using to calculate nitrosamines take into account the duration of treatment?
Yes. Yes, exactly. So
the second question. So going forward, what approaches have you taken to improve on the formulation methods, or rather the formulations of the products, so that we mitigate the formation of nitrosamines? And for those formulations that we have,
I think
are we required to request applicants to reformulate the products? Because I understand nitrosamine formation, the risk does not only come from the the excipients. There are some formulation approaches, for instance, for poorly soluble APIs. We understand they are quite notorious with regards to improving solubility. And one of the methods that has been adopted as a hot melt extrusion has been identified to be one of the causes of nitrosamine formations. So how, as the industry, do we aim at improving on manufacturing processes without really compromising on product quality as we tackle nitrosamines?
Yeah,
Okay.
so unfortunately didn't hear all portion of your questions, but the main thing is for standard nitrosamine there is a very defined acceptable intake limit which is going to be applied for all the medications, and that is for the standard small nitrosamine. They have the AI limit, whether it's in FDA, EMA guideline, etc. The compound specific AI limit that comes for the NDSRIs, which are, as the example I showed, and that's where you have to approach by the CPCA categorization, and that will give you a acceptable intake limit. So once you know that, then you can determine how to control that, by right analytical method, etc. So if you see that your limits are higher than the 10% of your target, it not necessarily it will immediately trigger a reformulation. So what you have to do is you may need some more, predictive stability calculation that can help in that case. You can put nitrite excipient scavenger. Those are also out there, for example, ascorbic acid and things like that. And that can help to actually reduce the amount of available nitrite to begin with. So that could be an easy fix for your formulation, for example. The other things is also when it's coming from manufacturing side, you have to have a better control on the utilities, on the water supply, for example, and other aspects like all the diagram. If you remember, I showed the sources of nitrosamine. Each will require a risk assessment done for those components, and that's how it needs to be done. It's a very comprehensive thing. A typical nitrosamine risk assessment could be anywhere from I have seen like anywhere from 80 pages more than 100 pages. So it's a very comprehensive thing that gets submitted at least on US FDA and EMA and other countries. Okay.
Thank you. Partha, I have a question. So
just type.
Can you please mute your microphone? If
Okay.
you're yes. So obviously new group of compounds, right, that are contaminating medicines, and everything seems to be still under development. You're still working out some of these things with the European Medicines Agency. And so, I mean, in the African context, what would be great to know is how do regulators and healthcare institutions, how do they protect patients given the limited testing capabilities, which you highlighted as one of the major, I guess, drawbacks in terms of how we are dealing with this issue at the moment. So what can we do in the meantime? Do we need to have a list of products that are on the watch list, that if you have these products, you need to pay attention, and what else should we be doing to keep patients safer?
Yeah, good point. So what I would say is, because of the limited analytical capabilities or in-house capabilities, what would be really good to see is if we can get hold of the nitrosamine risk assessment that was done by the marketing authorization holders for those imported medicines, which are notorious for knowing, which have known facts like ranitidine and all the obesity drugs, everything. There's 4 or 5 typical things, and they have very well established analytical methods and control. So we know exactly in these compounds these are the nitrosamines are present. So all you need to do without any analytical activity or capacity is get hold of those nitrosamine risk assessment package. If you take a look at that and you see that yes, they have done confirmatory testing before releasing those batches for import process, that's it. That will give you the tremendous confidence that you have something to begin with in a good shape. Then comes the other steps, that internally when your transportation done, is it done in the right way? So supply chain then kicks in for these import medicines, right? And the education for patients, are they leaving these compounds exposed to air or at not the right temperature? So that will also trigger formation of nitrosamine during the shelf life or the in use period of these compounds. So without doing anything, these are the things I can think of that will allow you to control. Now, if the marketing authorization holder is internally within Africa in a country, they have to go through this nitrosamine risk assessment process, and that means they will have to have the analytical capability or the instrumentation to prove that you don't have levels of nitrosamine on those batches. So it depends. Import medicine versus in-house manufacturing. That's how it will be. Yeah.
Thank you very much. Very practical places to start. Tebogo Ntoage, she's one of our analytical scientists on the line. I hope you took very good notes. There was a lot of content here for you. Come in with your question. Put on your camera, introduce yourself, your affiliation, and ask your question as briefly as possible.
Yeah, I can hang on for another 5 minutes or so.
Can everyone hear me?
Yes, we can hear you.
Hi.
Hi.
So my question here is with the nitrosamines, right? we've got a lot of products now that are on the market, and they are like old formulations. And if the studies were not done during formulation and API stage, can they still be monitored during the product life cycle?
Yes, they can be. And that's the whole purpose of the life cycle management, that you have to do either periodic testing, or if you have some idea that initially, let's say a couple of years back, they had the nitrosamine level of this, what you can do is take the worst case scenario, like a product which is close to its expiry date, and just test that. If you can do that, that will show you the realistic estimate that over the shelf life it has grown this much, this nitrosamine, and then you can draw a conclusion that, oh well, it looks like it has gone above the acceptable intake limit. Then it will require the trigger, every batch needs to be then monitored for nitrosamine. And if that is not an option, then you have to go back to the drawing board, unfortunately, and do reformulation strategy, or come up with better packaging, or come up with better awareness, basically, or a more sensitive analytical method. But I think realistically those are the options available.
Thank you, Partha.
Thank you.
We'll take one more question. Our speaker has to leave. And so Mary Nonde, you will ask our last question before the speaker leaves. So please put on your camera, introduce yourself, and ask your question.
Okay, I was with Mary Nolne. She has just walked out. There's an emergency that she has gone to, but she asked, does the risk or topical products weigh the same for oral and topical products? Do we have the same approach, or is it of concern if a product is to be administered topically?
Yes, okay. Partha, maybe just to help, Mary actually wrote her question in here as well. Let me just read it in case it clarifies what Lakson just said. So is it still critical to control nitrosamines in topical dosage formulations?
Yes, in some cases it's required, because they may have more local topical effect, and that will be more, I know, it's basically dosage form specific. So for example, if you have an inhalation product which has nitrosamine, or a IV product, that has more critical control that will be required versus a transdermal patch, for example. So in that case, the topical dosage form, since they are not getting that much locally absorbed, so the risk of nitrosamine will be lower. I would say oral solution is the highest, followed by solid oral dosage form, which is tablets and capsules, and then comes the topical dosage form. Okay, so you can have that control, but the general principles still apply, that if you do an analysis of the formulation and you see your limits are above the acceptable intake limit, you have to come up with a control strategy irrespective of dosage form. So,
Great.
Thank
you very much. Thank you very much. Thank you. I think, we will stop here. We want to thank our speaker for a really illuminating presentation, alerting our African frontline stakeholders of the challenge of nitrosamine. It's a new challenge. It's a rapidly evolving challenge. And in the African continent, we are especially vulnerable because we import 95% of our active pharmaceutical ingredients that we need to make medicines. We import more than 70% of the finished formulations, the medicines that people actually take. And that creates huge vulnerability. And the second biggest vulnerability that we have is the climate of high humidity, high temperatures, which is exactly the kind of conditions you need to create this risk. And what makes this very concerning is that it's affecting the major medicines that we're taking. For antibiotics, we have high rates of infections here, bacterial infections in children, and so forth. And anti-diabetics, we have a very high burden of that, antihypertensive. So it's really affecting some of our biggest high prevalence problems. And so it's a big issue. We face limitations in testing, in analytical capabilities, as well as in communication. Because of the regulatory challenges, we are not always able to communicate in a timely manner. And so big challenges. You have given us some recommendations of things that we can be doing without, as we build or as we put together these capabilities that we need to protect patients. And that's very useful. Things that regulators can do, NRAs, things that clinicians and healthcare professionals can do on the front lines already to keep patients safe. Awareness of recalls and alerts that are coming from US FDA, from the European Medicines Agency, because they're following this issue closely, and we're taking the same medicines, medications on the African continent. So those things will apply. Communicating risks and benefits to patients, and of course, making use of therapeutic alternatives when you're facing perhaps a product that is vulnerable to these kind of challenges. So thank you. Thank you very much for giving us some real tools that can now immediately be used to protect patients, supply chain actors doing proper due diligence in supplier selection, having traceability, using good storage and distribution practices. All of these things will help us to mitigate. We're not going to remove it, to mitigate and minimize the challenge of degradation that results in nitrosamine contamination in medicines that patients on the African continent are relying on to get better. And so, first of many conversations, Partha, we would invite you to come back. Let's have a discussion, you and I, after this, and we can think about doing perhaps a more targeted trainings and webinars that is targeting the right audience. You presented us a lot of beautiful chemistry, and maybe we did not appreciate it as much because we are not analytical chemists, but we'd like to bring this to the attention of the right people because it's important, it's needed, and it's what we need to do to make patients safer on the African continent. So thank you very much for coming this evening, for sharing all of this. And thank you to our audience and participants for hanging on and, delaying dinner and asking great questions. We will do a recap of this in the next webinar. And yes, it's a conversation. We'll keep talking. Thank you, Partha.
Thank you, Lenias, and thank you all. And I really look forward to partnering with all of you, as I'm willing to help out. And, chemistry, it's all chemistry to begin with, how nitrosamines are forming. So it's the boring part, but you have to know the basics. And main thing is to keep in mind, my last note is medicines are only contributing 25% of daily intake of nitrosamine in a human body. That's the very key thing. Food, water, etc. are contributing to 75% of the nitrosamine getting ingested in the human body on a daily basis. So we are just looking at a very small fraction of the whole picture. So keep that in mind. We're talking about pharmaceuticals. That's why it's a small portion, but the bigger picture is much wider, like water, the food, everything is actually contributing to nitrosamine. So something to discuss maybe later on.
Absolutely. Absolutely. We'll bring you back and we will have more detailed conversations longer with more time for sure. Thank you so much, everybody. Have a great day and good evening. We'll see you next time. So the next webinar will be in 2 weeks. And it's going to be a case study across 10 hospitals in 4 countries. So it's going to be a super, super interesting one. And we hope to see you then. Thank you very much.